So, very good morning, friends. We all know that the relation between CKD and GI microbiota is an emerging area of investigation. GI microbiota refers to trillions of population of microorganisms. In chronic kidney disease, an overall decrease in diversity is reported. With the advent of high throughput sequencing techniques, the entire GI microbiota community, as well as their function and interaction with the host, can be studied. To characterize the GI microbiota in various stages of chronic kidney disease, and then dietary supplementation for manipulating the resident population in a beneficial way, we are having with us today Dr. Gopal Basu, who, after completing his specialty training in 2008, progressed to serve as a professor of nephrology at prestigious CMC Vellore, India, before migrating to Australia in 2015. Presently, he is lead transplant nephrologist at the Alfred Hospital and associate professor at Monash University, Melbourne, Australia. He is a keen nephrology educator and is an active member of International Society of Nephrology Education Working Group, Social Media Education Subgroup. He is one of the founder members of the ISN Education. He had won several awards, including the Distinguished Educator Award by Transplant Society 2013. He was also recipient of the Tanker Award in 2009. So he is having very illustrious career, and I request Dr. Gopal Basu to deliver his talk on microbiota in CKD and how to manipulate in a beneficial way. Dr. Gopal Basu. And after his speech, my co-chairperson, Dr. Alens and Dr. Arun Kumar will conduct the question answer session. Thank you.
Thank you very much for that generous introduction, Dr. Om Kumar, and thanks to the organizers, especially to Dr. Narayan Prasad and my esteemed colleagues and Chennai. I would have loved to have been there in person to give this talk, but unfortunate point in time. I won't waste too much time, and I'll go ahead with the talk. I'm going to share my screen now. So we're going to look a little into gut microbiota and kidney disease, and there seems to be an emerging field, and there's a lot of interest in gut microbiota, and we're able to discern this a little bit more clearly because of the availability of techniques to study gut microbiota. So before we go into the talk, we should define a few things. Gut microbiota and microbiome are different. Microbiota refers to the 100 trillion microbial organisms that are there in the human gut, and they are established at birth and modified based on several factors. The gut microbiome refers to the combined microbial genome of the gut microbes. And from these 100 trillion microbes, there are about 3.3 million prokaryotic genes in the gut microbiome that are unique. And a study of the collection of genomes from a mixed community of these organisms together is called metagenomics. We're able to study this gut microbiome at a metagenomic level because of advanced tools that are available, like next-gen sequencing of 16 sRNA and whole genome sequencing. So the gut microbiota, the huge population of bacteria that is present, can be considered metabolically active endogenous organ, and it's defined by its composition, its dynamics, and stability. So to analyze gut microbiota, you usually source the stool sample and look at the microbial cells, either in a culture or put through a cell sorter, or simply look at the microscopy. Those are the ancient techniques that we used to have. We could also look at metabolites using mass spectrometry, and nucleic acids are now being screened using amplicon-based survey assays, as well as next-gen sequencing. Now, once you get the microbial level genomics, you can then study that for the whole gut microbiota, called that becomes metagenomics. If you study the transcriptomics, it's called metatranscriptomics. Then you get the output from these transcriptome processes, that's the proteins. So if you study all the proteins that are present, that's called metaproteomics. And then you have the metabolites, the enzymes that act and produce some metabolic end products. Now, this then be integrated with the host characteristics, such as the host gene expression, the clinical information of the host, and you get the metadata from that. Now, this can then be used to, in different study circumstances, you could do case control studies, you could do studies based on a therapeutic intervention before and after, or you could even do a quantitative analysis of how much a protein gene or a metabolite is expressed or not. And once you figure that out, then you find out a candidate gene or an intervention, and then you try and apply that, take it back to the lab, and use an animal experiment to kind of prove whether that intervention makes a difference or not. So there are lots of steps in understanding how gut microbiota affect human organ systems, including kidney disease, and how we can go about finding a particular tool to help us intervene. The gut microbiota, as I said, is established in newborn. There are several events that take place during growth, from infancy, early childhood to adulthood. The various events, including diet, environmental exposure, illnesses, medications, physical activity, and all, and the community as such. All of that would actually affect the microbial, the gut microbiota in the adult. The microbes in human, these 100 trillion microbes in the human body remain in symbiotic relationship with the human. So when the gut microbes remain in symbiosis, we find that there are about 8 to 10 varieties or types of microbe bacteria that are present, which account for about 800 species, and together they constitute about 1,000 milligrams of endotoxin in the gut. And the Human Microbiome Project in US collected microbiome from several participants, and they looked at it, and they realized that there is a wide variation in the microbiota. While and this is not a particular bacteria, it's actually a big phylum of the bacteria that we're talking about. And you can see that there are two major groups, the bacteroids and the firmicutes, and there's a wide variation between individuals, even though they are healthy. And for each person, the symbiotic relationship remains stable until something changes, like maybe diet or exercise or hormone or stress or environmental factors or disorders that changes this symbiotic relationship into what we call as dysbiosis. And this is seen in several illnesses and is attributed to be causatively or at least associated with several of these illnesses. The interaction between the microbiota and host is interesting. Microbiota interact with the host. There's a bacteria to host kind of interactions. The bacteria can result in several advantageous relationships with the host. The host also provides several advantages to the bacteria. And there is also interaction between the bacteria. For example, if you have commensal bacteria, they do not, or they inhibit the growth of pathogenic bacteria in the gut. So while the microbiota benefits from the human, and the human also benefit from the presence of microbiota by not only pathogen control, but also, biosynthesis of several vitamins, amino acids, cardiovascular health, and immune maturation, etc. So how do we coexist with these microbiota in the gut? So you've got the intestinal epithelium here, and you've got a thick layer of mucus there, which has got several of the molecules that form a barrier between the commensal bacteria that are sitting outside here and the human gut tissue. And there are four important components of this barrier. So one is the mucus that is being secreted by the goblet cells. Then you have the intestinal epithelial cells carrying the toll-like receptors, which are pathogen recognition receptors. And then in the mucus layer, you have antimicrobial proteins, and you also have secreted IgA from the plasma cells that is present in the mucosa that kind of prevents the bacteria from directly entering the intestinal mucosa. And these barriers, underneath these barriers, you've got the intestinal epithelial cell, and then you've got the lamina propria, which has got a wide variety of immune cells. And you know that in the small intestine, these immune cells can be organized into pairs patches. Now, commensal gut microbes maintain the gut functional integrity as well. They help maintain this functional integrity by various mechanisms. Upregulation of mucin genes that produce mucus, restoration of the tight junctions between the intestinal epithelial cells to provide a barrier. They also help secrete the antimicrobial proteins, and they suppress the intestinal inflammation, and so on and so forth. Now, the gut microbiota also have an important role in immune regulation. They, as you know, are important in immune education in infancy and in childhood. They are also, the commensals form a biological barrier against pathogens colonizing the human gut. And they also regulate the intestinal epithelium by producing short chain fatty acids, which are very useful to maintain the intestinal epithelial integrity. So this functional barrier is where the bacteria is, and because there's a lot of these bacteria, these commensal bacteria, they continue to produce the commensal microbe-associated molecules such as lipopolysaccharides, which are constantly triggering the toll-like receptors here, inducing what is known as a tolerogenic immune response, whereby the dendritic cells trigger the T cells to become T regulatory cells, and therefore there is a suppression of inflammation that happens, and this has a systemic impact as well in terms of reducing inflammation overall. The short chain fatty acids are breakdown products of dietary fibers. So dietary fibers are broken down by microbiota to produce these short chain fatty acids, which then have several functions, including proliferation and cell metabolism, maintenance of the intestinal epithelial cells, maintaining the barrier function, endocrine functions, and gut motility, as well as immune regulation. So these short chain fatty acids that are produced by the microbiota that are present in the intestine from the dietary fibers help maintain the intestinal integrity as well. Now, this is symbiosis. What happens in dysbiosis is that there is a change in composition or diversity of the microbiota, and that results in change in metabolites that are produced by the microbiota. And often, as you can see, there's an intestinal epithelial barrier is lost, and there is translocation of not only the complete bacteria that happens in extreme cases, but also the bacterial end products, which spill over and cause systemic inflammation. So as you can see in dysbiosis, there is a lot of pathobionts that kind of break through the barriers that are present. So there's a loss of tight junctions. There is a production of a lot of pathogen-associated molecular patterns, which are often triggering inflammatory response. And here the dendritic cells, because of the pathobionts, they are new and they're not usual common cells. The dendritic cells react to produce T helper 17 kind of cells and not the T regulatory cells, and therefore there is actually heightened inflammation. And that inflammation again reduces the mucosal integrity and also spills over into circulation to reach inflammatory signals throughout the organ systems, including the kidney. And this inflammatory signals in the form of cytokines. So you have IL-22 that is reduced, and IL-17 and IL-17F result in neutrophil recruitment. And all of this result in a pro-inflammatory cytokine response. So what happens in CKD? Does gut microbiota and CKD interact? How do they interact? So gut microbiota results in change in composition and diversity of microbiota. It results in microbial translocation, as we saw. There's a disruption of the epithelial barrier. And not only that, the pathogenic microbes would produce the increased amount of uremic toxins from the dietary protein. And it results in systemic and kidney effects as well. There is immune dysregulation systemically, causing renal inflammation and a trigger of fibrosis. There is also a trigger for hypertension and infection-associated kidney diseases. So let's start with what happens to the gut microbiota first. So as you see, in various kidney disorders, microbial population has been studied compared to general population microbiota. The general population is indexed from national level microbiome projects, such as the HMP project in US. And you can see that because there's a wide variation, even in normal individuals, it's quite difficult to make sense of what really happens. And you only have generalization of which phyla or which particular group of bacterial colonies is more or less in a particular situation, such as, for example, the chronic kidney disease, you get more bacteroids and less firmicutes. And in end-stage renal disease, again, you get more firmicutes and not so much bacteroids. So there are lots of changes which are difficult to make immediate sense of because of multitude of influences that happen here. Even in nephrotic syndrome, there seems to be change of gut microbiota. There is some association between the clinical features such as low serum protein and proteinuria with presence of certain microbiota in some small number of studies. And if you look at studies like that for different kidney diseases, for be it idiopathic nephrotic syndrome, membranous nephropathy, MPGN, or IgA nephropathy, you do see that there's lots of various different kind of bacterial populations that upregulated in the gut microbiome, and some others are actually downregulated in the gut. And same applies for conditions such as hypertensive nephropathy and diabetic nephropathy. There is a lot of variation, and scientists are in the labs trying to make sense out of what these variations are and how do they make sense in terms of clinical features and are they related or not. So chronic kidney disease can facilitate gut dysbiosis because in CKD, there is change in diet, there's intake of numerous medications, and also because the kidney is not producing or excreting urea, there's some amount of urea and nitrogenous waste excretion through the gut. All of these result in several ways in which they change the gut microbiomes. As you know, what you eat helps modify what the microbial population remains in the gut. So if you eat a very plant-based diet, there's a different microbial population compared to an animal-based diet. And depending on medication, so for example, if you're on proton pump inhibitors, then it changes the gut microbiota. And we know that because proton pump inhibitors have been shown to increase Clostridium difficile infections, and that is simply by alteration of the gut microbiomes. Similarly, the nitrogenous waste products and urea that are secreted in the gut can result in selection of urealytic agents in the gut, similar to and Enterobacteriaceae and Enterococci are upregulated as well. And all of these result in immune dysregulation. There's also mucosal edema, gut mucosal edema with fluid overload, and a wide variety of reasons for constipation, all of which can result in dysbiosis. So chronic kidney disease can itself cause dysbiosis. But CKD, uremic toxins impact the gut microbiota as well, the secretion of uremic toxins, and it results, as I said, an increase in Enterobacteriaceae species and a decrease in the protective Lactobacillus and Prevotella species. The gut dysbiosis in turn also facilitates chronic kidney disease, and that happens by a multitude of mechanisms. As you know, that dysbiosis results in an increase in more inflammogenic species, and there is a lot of translocation of lipopolysaccharides and bacterial antigens causing systemic inflammation. And that results in also an increase in certain kind of bacteria which have these enzymes, the urease, uricase, tryptophanase, or precusol producing enzymes, which tend to break down protein-based diet into a lot of uremic toxins, which are then absorbed in circulation. And then not only that, there is a reduction in short chain fatty acid producing bacteria such as lactobacilli and prevotella. And this results in loss of mucosal barrier in the gut and therefore further inflammation. And a byproduct of this inflammation and is also an activation of sympathetic nervous system and vagal system, causing evidence for hypertension. So the dysbiosis results in inflammation and fibrosis in the kidneys by systemic inflammation. There is also local activation of gut-associated lymphoid tissue. So there is increased production of IgA that is triggered by these dysbiosis, and thus can be attributed to what is seen in IgA nephropathy. High protein and high cheese containing diet have a lot of amino acid precursors for uremic toxins, which in the presence of the dysbiotic gut can result in production of indole, phenol, p-cresol, and trimethylamine. All of this is then modified in the liver to form the uremic toxins, which then reach circulation. These are usually cleared by the kidney, but in the presence of CKD, these accumulate in the circulation, and they result in several systemic impact. What are the described impacts include renal interstitial fibrosis and glomerulosclerosis, arterial vascular calcification, endothelial dysfunction, endothelial leakage, renal inflammation in general, reduced erythropoietin sensitivity, as well as in several studies has been associated with increased mortality. And as I said, the precursors for these uremic toxins come from animal-based proteins, as you can see here. And each of these components, including precusol sulfate, has been associated with RAS activation and CKD progression. Indoxyl sulfate is associated with TGF-beta inhibition, reduced podocyte viability. Trimethylamine oxide has been associated with SMAD3 phosphorylation, collagen deposition, and interstitial fibrosis and mortality. And there are several cross-sectional observational data from humans which have shown association between higher levels of uremic toxins and progression of chronic kidney disease and association with interstitial fibrosis and even cardiovascular morbidity and mortality. Not only is uremic toxin accumulation or production from the gut dysbiosis important, it's also the fact that there is systemic inflammation and activation of an inflammatory pattern from the gut dysbiosis that causes further signals to interstitial inflammation and fibrosis. But it also increases the risk of proteinuria. So gut inflammation can cause proteinuria either directly by causing pathogenic IgA nephropathy or amyloidosis, or T regulatory activity upregulation, and diabetes and RAS inhibition. Gut dysbiosis is also associated with diabetes and hypertension and renal calculi by oxalate metabolism inhibition and so on. So there's some multiple ways in which gut has dysbiosis results in chronic kidney disease or progression. Now, for therapeutic interventions, there is very little that is available in terms of information or data. Most of this is animal studies. And there are lots of review articles on various opinions based on small studies. They talk about how this could be an important factor to consider. So you could have, dietary changes or exercise can actually modulate gut microbiota. And then specific medical interventions to modulate gut microbiota, including probiotics, which are live microbes, prebiotics, which are food ingredients, symbiotics, which are a combination of these two, as well as specific medications. Plant-based diet, as you know, is helpful in retarding chronic kidney disease progression, and it seems to promote short-chain fatty acid producing bacteria in the gut. And several studies, including the Singapore Chinese study of 63,000 adults and studies elsewhere in Belgium and in US, have shown that plant-based diet tend to reduce the progression of chronic kidney disease and possibly through gut microbial alterations as well. Physical exercise has also been associated with improved gut microbial population and a reduction in pathogenic gut microbiomes. And animal studies have shown that lactobacillus supplementation can improve exercise tolerance by the animals. Now, there are lots of animal model studies that have proven impact of uremic toxins and how blocking uremic toxins by substances such as dimethylbutanol have reduced not only renal interstitial fibrosis, but also hypertension and proteinuria in animals. So be it interventions including high fiber diet or directly replacing short chain fatty acids such as acetate and butyrate, modifying the gut microbiome using SGLT2 inhibitors have also shown to improve renal function and reduce the uremic toxins. There have been studies that have looked at blockers of certain channels to increase gut motility that have also shown to work in animal models. And replacement of lactobacillus in sham nephrectomy, sham as well as 5/6 nephrectomy models have shown improvement in renal function as well. And similarly, fecal microbiome transplantation in mice have also shown to be of some use. But all of these studies have taken place in animals. In humans, there are very few far
Sir,
studies.
Sorry to interrupt last minute, sir.
Yeah. So these have shown that variably that there is some benefit in using probiotics, but this is neither reproducible nor are these studies show a significant impact. Fecal transplantation is also is in infant stage. There's lots of risks and the safety concerns about it. So while there are lots of things that are portended to be beneficial, there seems to be very little that is proven to be useful. And more importantly, the Indian gut microbiota is not very clearly known. As you can see, there's wide variation even in normal healthy gut microbiota from various states of India. So to make sense of what a CKD gut microbiota in India would be compared to the Western data is going to be difficult because of the variation in the diet and the commensal bacterial population. So there's lots of questions. It's an interesting area, but there's lots of questions that need more answers. Progress in therapy and development has to move from the lab back to animal experiments, finally to clinical trial before it comes to clinic. At the moment, there is some light at the end of the tunnel. It could be a way out, it could be an oncoming train, or it could just be a humbug. We have to wait and see. Thank you. I'm happy to take any questions.
Yeah, thank you, Dr. Gopal, for excellent opening of the today's session, because it has got huge financial implications on the patients of CKD. Two questions have come in the chat box. One is from Sam Bansal, whether it should be recommended in each and every CKD patient. If yes, then what should be the dose? Second question is from Dr. Pradeep. Yes, there is a lot of data in prescribing prebiotics or probiotics in CKD, whether there is any data of their use in AKI also.
So in the first question, I wouldn't be recommending specifically using probiotics in CKD yet. There is no clear data to recommend that, even to a significant amount of evidence to say, yes, there may be some benefit in human studies. That is not, other than reducing uremic toxin levels, there's not been a direct relationship with any clinical outcomes. So I wouldn't recommend probiotics as yet, but it doesn't, it's very easy to modify your diet to plant-based diet and do exercise, and that might even do the trick in terms of changing the gut dysbiosis in general, and those are interventions that are easy to undertake. As to the second question about the data of supplements of pre and probiotics, I think all the CKD progression delaying studies are all from animals. There are no human studies that have clearly shown that the CKD progression is retarded by use of pre or probiotics. So that is contentious as such. And there are animal data of ischemia reperfusion injury in mice where use of short chain fatty acid carrying bacteria can improve renal function faster. But there's again no data in humans. So we are talking about something that is still in the lab, which is still not yet been refined enough to be even to have a clinical trials in large in a large setup. So it's far away from the clinic at the moment.
So thank you very much, Dr. Gopal. And with this, we come to the conclusion of this session. I thank the organizers, the ISN and Secretary Dr. Narayan Prasad for giving us opportunity. Thank you, Dr. Gopal. And the session is handed over to the event manager.